Good afternoon, everyone. Welcome to the 8th Dialogues in Biological Anthropology. My name is Jeremy De Silva. I'm an assistant professor in the anthropology department here at BU, and I'm incredibly pleased to introduce an amazing panel of discussants. This is like for me, it's like maybe a first-year shortstop at my first All-Star game, looking around at the future Hall of Famers. To my far left is the director of the Institute of Human Evolution at Arizona State University. That's Dr. Bill Kimball. To my immediate left is the department chairperson at the Department of Anatomical Sciences at Stony Brook University, Bill Jungers. And to my right is Matt Cartmill, the department chair of the anthropology department here at BU. And we're discussing today an absolutely amazing fossil discovery that was made in the 1990s and published in 2009 in the pretty extensive issue of the journal Science. And that, of course, is the Ardipithecus ramidus skeleton. Ardipithecus ramidus is one of many new incredible discoveries that have been made in the last couple of years. This is a wonderful time to be a paleoanthropologist. And it's quite an amazing time to be a student of paleoanthropology as well, I hope, for those of you who are here willingly or here because you got some bonus points on your final exam. I'm not sure yet. But we're going to talk about this skeleton today because any new discovery, any amazing new discovery like Ardi is, comes with controversy and comes with important discussion. And we're going to hope to flesh out some of those issues today. But before we dive into that, I want to provide a bit of perspective on this and how esteemed our panel is. In the mid-1970s, when the very famous Lucy skeleton was discovered at Hadar by Don Johansen, in the years that followed, some young scholars had an opportunity to study that skeleton and study the material from Hadar and interpret what they were seeing in the craniodental and postcranial remains. And of course, craniodentally, Dr. Kimball, and postcranially, Dr. Jungers made incredibly important contributions to our understanding of what was at the time the oldest human relative skeleton in Australopithecus afarensis Lucy. And now we have a new potential oldest human relative skeleton in Ardipithecus ramidus 3 decades later. And so it's really something special to have these folks here talking about this skeleton. So the way I want to start off is just have Drs. Kimball and Jungers explain a little bit about their position here, where they're coming from in terms of their own interpretations of Ardi, perhaps where they started and now where they've maybe not ended, but where they are currently in understanding Ardi, and then for a different viewpoint on the interpretation of the skeleton, an important viewpoint, Professor Cartmill is going to speak for a few moments. And then we're going to answer some questions that all of us have up here amongst ourselves, and then we're going to open it up to questions from you guys, and hopefully you have plenty of them. So I'll start with Dr. Kimball.
Thank you, Jeremy. Good afternoon, everyone. My starting position is as follows. If you take a human skull and you compare it to that of an ape and turn it upside down, look at the base, which you might think of as the invisible anatomy, part that you can't see when you look at a whole skeleton unless you search. There are an enormous number of differences that relate, some to posture, some to the shape and size of the brain. Most obviously, the position of the foramen magnum, the great opening on the base of the skull through which the spinal cord passes and flanked on either side by the occipital condyles where the vertebral column articulates with the head, is very far forward and central on the base of the skull in humans compared to almost any other primate. And in the process phylogenetically of the foramen magnum moving forward on the base in humans, the neurovascular channels, which are manifest on the base as foramina openings, spread out so that in humans the central part of the base is not only very short with the foramen magnum very far forward, it's indeed in the center of it very wide in relation to the overall size of the skull. We see already these changes and other associated anatomical modifications that are associated with them in the temporal bone and the occipital bone. We see them already in the earliest Australopithecus skulls at 3.5 million years ago or so. So the question is that given that those changes tie Australopithecus and all hominins subsequent to us, does Ardipithecus have those same modifications that are associated with its relatively abbreviated cranial base and forward foramen magnum, as was first described in the journal Nature in 1994 when Ardipithecus was diagnosed as a relative of ours. And the work that I've begun with my colleagues Gen Sua, Tim White, Yoel Rak, and Berhani Asfaw shows that in fact Ardipithecus does indeed have many of those additional modifications, including a very wide central base and other morphological accommodations to the shifting around of these landmarks. As the foramen magnum moves forward, the structures move out to the side, the neurovascular structures move out to the side. It would appear that these changes are part and parcel of an adaptation to becoming upright. And as Bill will speak more directly to, they seem to tie what's going on to the base of the skull, in the base of the skull, to what's going on elsewhere in the body, and makes Ardipithecus, despite all its oddities, and there are plenty of them, makes it a relative of yours and mine, and less so to that of a chimpanzee or a gorilla.
Thanks, Bill. And now for a postcranial perspective, turn to Bill Jungers.
Yeah. Well, my position has been a little bit of a moving target. When Artie first appeared, I was certainly agnostic, and I think I was overwhelmed by the foot, the arboreal grasping signal in the foot, and sort of missed some other more subtle, but I think diagnostic features. I think there aren't many features that are diagnostic of bipedality in Artie, but I think there's some very good ones, and that includes the lateral digits, the joint between the metatarsals and the phalanges. Again, I was also rather skeptical about the pelvis when it was first published, in the sense that I couldn't see how anyone could make sense out of that puzzle. But having a chance to see it, it became clear that there really was a part of that pelvis that appeared to speak to bipedalism. That is the very short iliac height. And so suddenly you had a pelvis that had features of the false pelvis that looked hominin-like, connected to a true pelvis that was decidedly primitive and ape-like. Some of the other features that have been reconstructed, I think for now I have to take on face value, and I think many of us may have to make our own judgment calls about it, because I don't think it's likely to happen in the near future that people are going to have the opportunity to methodically take every little piece apart and put it back together. But if the reconstruction is correct, there are some interesting things about iliac blade orientation that also is compatible with bipedalism. And then I was very pleasantly surprised by the recent paper by Rousseau and Kirk that seemed to link the position of the foramen magnum not just to orthograde posture, but in hominoids in particular, specifically to bipedality, and that seemed to be characteristic of bipeds and other kinds of mammals, and I thought that was fascinating. And so I saw these features, not numerous, but sufficient to diagnose bipedality, and they could be added to the character list of features that are synapomorphies between Artie and other hominins. And finally, just I mentioned the hand. It is a powerful grasping organ, but it has quite a reduced thumb. The phalangeal curvature is really extraordinary. And I think that there are other signals in that hand that indicate that it is not the hand of a quadruped. And it's certainly not a quadruped that in the kind of categories we normally define. It's not a knuckle-walking hand. It's not a digitigrade hand. And contrary to what I thought I read in the science papers, I don't think it's the hand of a palmograde quadruped either. And you know, I think Matt made some good points. The hand is long relative to the foot. The phalanges are very curved. This is a hand that's capable of a lot of things, but I don't believe it's the hand of a quadruped.
Thank you. So many students, I think, that are first learning about human evolution, first learning about Ardipithecus and its place in human evolution, don't necessarily see the game-changing ideas that were presented in the paper, because as you go back in time through genus Homo into Australopithecus into Ardipithecus, of course you're going to get something that is not as good at bipedalism and maybe just shows these subtle changes to bipedalism, like the foramen magnum and the lateral metatarsals and things like that. And you're not going to have as large of a brain size, so what is the big deal? Wouldn't we have expected to find something like this? And the important thing to recognize is that the interpretation of the skeleton is that the body plan of this thing is generalized in a fashion that all of the modern apes would have had to have independently evolved their suspensory orthograde body postures and locomotion. And so I will turn to Matt Cartmill to talk about some potential problems associated with that interpretation of this generalized body plan.
Well, thanks, Jeremy. I'm going to talk about this very briefly because I am mindful of the cautionary principle that Bill Jungers put up on his last slide, which is if you haven't seen the fossils, shut up. And I haven't.
Not quite that.
Well,
What you meant.
I'll put it more kindly. Before speaking, it is always best to have seen the fossils. And I haven't. But I have been reasonably careful about this. I haven't shot off my mouth ignorantly in print. I've only shot it off ignorantly at public conversations and talks like this one. So I'm going to do it again, but I'm hoping to see the fossils before I actually have to write something about it in the next edition of my textbook with Fred Smith. But at any rate, I haven't come all the way into the fold the way that Bill Jungers has. From being the strayed lamb. But I've come all the way up to the gate, and I'm just sort of standing outside, bleeding resentfully. And so let me do a little of that bleeding for you guys now. When I saw the Ardi papers in that special issue of Science in 2009, I was indignant. I looked at the cover photograph. Let me rehearse the claims that are being made here. The claims are, one, this is a hominin. That is, it's more closely related to human beings than it is to our nearest living relatives, the chimpanzees. Secondly, it's an ancestor of modern humans. Specifically, it evolved into the first species of the genus Australopithecus, Australopithecus afarensis. And thirdly, it shows no signs of having ever passed through an ape-like pattern of locomotion or adaptations to an ape-like suspensory, upright, orthograde, arm-swinging kind of locomotor adaptation. In fact, the last common ancestor of humans and chimpanzees, according to that paper, according to the papers in that issue of Science, was an above-branch quadruped, an animal that walked around on top of branches like a monkey. Okay, I had problems with all of these things. I looked at the cover photograph of the skeleton, and I said, wait a minute, this thing, it says here this thing has forelimbs that are the same length as its hind limbs, but it hasn't got a humerus, and it's only got a little chunk of femur, and so the length of the upper arm and the length of the thigh are being estimated from the length of the radius in the forearm and the shin bone or tibia down in the calf, and if you look at the photograph on the cover, it's obvious that the tibia is shorter than the radius. So how do you come up with the same length for the fore and hind limbs? Something is wrong here. And I didn't believe a whole bunch of other stuff. So I did, however, go and look at the casts. These are good, high-quality casts that Owen Lovejoy was kind enough to let me come and look at Kent State University in Ohio. And like I say, I didn't come all the way into the fold, but I came to appreciate that a lot of the things he was saying were true. The fore and hind limb elements that are preserved really are about the same length. I don't know what happened to the photograph of the tibia on the cover of Science, but the tibia really is about the same length as the radius. With the worst and most evil intentions in the world, I could not make that tibia significantly shorter than the radius, no matter how I fiddled the pieces and so on. Okay, couldn't do it. So I'm willing to accept, I think, a lot of the same conclusions that Bill Jungers was driven to, perhaps even less reluctantly than I was. I think this is an animal that is probably on the human, not the chimpanzee branch of the African ape tree. That is, it's a phyletic hominin. I think it probably was spending a certain amount of time walking around on its hind limb. There are adaptations in the hind limb, like the doming of the metatarsal heads that Bill Jungers talked about, that really are pretty distinctive of and diagnostic of the kind of terrestrial bipedality that we see in human beings. That is, walking around, flapping your forelimbs in the air and pushing off with your hind feet. So I'll buy into it that far. What I don't buy into, I still don't buy into, is the proposition that the last common ancestor of humans and chimpanzees was an above-branch monkey-like quadrupedal animal. And those of you who saw the webcast at least saw my typographical display of petulance, in which I put up about 50 supposed, well, not just supposed, real synapomorphies, non-primitive features that all the living apes and humans share in common with each other, and most of which, in fact, all of which are reasonably interpreted as having originated as adaptations to arm-swinging, suspensory kinds of locomotions, brachiation broadly defined, including the sorts of things that orangutans do. Now, that said, I think Bill Jungers is right to say that some of these things we know predated an ape-like postcranial morphology. For example, tail loss. Okay, the loss of the tail, I think, has something to do with the reduction of galloping and rapid quadrupedal above-branch kinds of gaits in the ancestral hominoids, but that doesn't necessarily imply vertical posture. Maybe that loss of the tail was pre-adaptive for vertical posture because it freed up the tail muscles to become the pelvic diaphragm that helps to support your pelvic entrails and keep them from falling out between your legs in a vertical posture. And maybe it does that job and evolved that job independently in orangutans and gibbons and chimpanzees and humans and so on. Fair enough. But there's a lot of other stuff about the human and ape postcranial morphology that doesn't really make sense as having originated in any other context other than a whole lot of use of the forelimb to lift and move and propel the body, more so than you would see in any kind of quadruped. Just a couple of morphological examples. In human beings, the latissimus dorsi, that's that big muscle, the lats, for those of you who work out, which I don't, that's that big muscle that forms the back wall of your armpit. You stick your fingers into your armpit, the latissimus dorsi has the massive meat in back of your fingers. It pulls the humerus downwards and backwards. Obviously an important propulsive movement. Okay, and it's an important propulsive muscle in a quadruped too. But in quadrupeds, it attaches only to the vertebral column. In human beings and apes, it goes down and attaches to the pelvis, so that it lifts the pelvis by downward movement of the upper limb. This is not useful in, and it's never found in, quadrupeds. Pectoralis minor, a little muscle that comes off the rib cage in front and goes up to the upper arm in most mammals. In humans and apes, it only goes to the shoulder blade. Okay, so it doesn't affect the independent movement of the upper limb on the shoulder blade. It just helps to support the entire body through the scapula, through the shoulder blade. And so on. There are, 40 or 50 little details of human and ape anatomy like this that, to my mind, are giving us a signal of suspension, of orthogrady. And I see that even in the Ardipithecus skeleton, as Bill Jungers mentioned. This is one of the striking things about the Ardipithecus hand and foot is that the hand is a lot bigger than the foot. And the data shown and given in the science paper in 2009, if you look at them carefully, you'll see that in this respect, Ardipithecus sorts with brachiators. It doesn't sort with quadrupeds. It doesn't sort with bipeds. It sorts with the African apes. It's got a big hand and with big, highly curved phalanges and a rather dinky little thumb. Okay, this tells me that this is an organ that is being used to a considerable extent as a hook-like structure for pulling upwards, as opposed to the foot, which has this big, robust first metatarsal sticking out. That tells me that this is a grasping organ, not used as a hook at all. In the trees, it's presumably grasping the thing that the animal is standing on. So I see signs in the Ardipithecus skeleton itself of the kind, some adaptations, at least in an early and incipient Miocene ape kind of way, to the sort of orthogrady and suspensory locomotion that Owen Lovejoy and Tim White and their co-workers would like to read out of the human lineage altogether. I remain skeptical about that, and I still need to be convinced. But maybe I'll make my way into the fold eventually.
I would say that a little bit like poison, it's sort of, pick your homoplazy. I think that there are features of the pelvic and proximal femoral anatomy of other hominins, like Auroran, that do, and even if you throw Auroran out and just compare some of the Miocene apes, earlier Miocene apes to later hominins, it's pretty clear that the phonetic resemblance, at least, the shape similarities are much greater between those proximal femora and hominins than they are with any of the great apes. So in that final plot that I sort of threw up from a recent paper, it looks to me like, at least in some aspects of their anatomy, they are homoplastic, that they are features that have evolved convergently. But I'm also sympathetic to the notion that some aspects of the bowel plan are hard to explain from an ancestral quadrupedal position. But I do think that there is rampant homoplasia in any way you want to connect the dots.
Yeah, and I would just amplify that point, and I think Matt gets it, but not everybody else necessarily does, because it's a relatively subtle point, that the rampant homoplasia is not dependent on Ardipithecus being more closely related to us than to a chimpanzee, for example. Because if Ardipithecus is in fact, granting the argument, primitive for the large-bodied hominoids, you have the same problem with homoplasia no matter where it's rooted within the hominoids, whether it's a relative of chimps or humans or gorillas or what have you. It's the same problem. So these are very different questions and draw on different data for each.
Yeah, I agree. Let me put that a little slightly differently, which may be easier for some people to understand. Suppose Ardipithecus was a primitive gorilla and showed signs of having a quadrupedal ancestry. It would be the same quadrupedal ancestry that it would have to be if it was a primitive chimpanzee or a primitive human. That is the last common ancestor of the African hominoids. So no matter where you stick it on the branching diagram, as long as it's giving a genuine quadrupedal signal, it poses the same problem.
Yeah, correct.
Should we open it up for questions? Hopefully. You have questions? All right, if you do, raise your hand and a microphone will come around to you.
If you have answers, also raise your hand.
Yes, please.
Yeah, we'll take a few of those.
Can we ask questions of other experts in the audience?
Yeah, I don't see why not. We do have experts in the audience.
There's certainly a couple of people in the audience who know a great deal about pelvic anatomy, people in the audience who know a lot. I'd be curious to see what some of those people might think about the Ardipithecus pelvis.
Are we asking this question?
I am.
All right.
I don't know. Maybe everybody will answer.
So if folks don't have, do you guys want to speak to this? Do you have thoughts on the pelvis? Sorry to put you on the spot. Either of you, actually. We have here Dr. Warner and Dr. Luton.
I am interested to hear your discussion and interpretation of the pelvis. I, like you, was skeptical when I first saw the pieces that it began as, and then the final whole in terms of the reconstruction. And I find myself wavering back and forth whether or not how convinced I am. I agree with you that the shortening of the iliac blade is probably something real that we can see. I'm less convinced about the rotation, and I am less familiar with the attributes of the lower part of the pelvis. If you could speak to those for a minute in terms of like hamstring attachment or so forth, in terms of what you see there, I would be fascinated to get your perspective on those issues as well.
Please.
I also haven't seen the fossil, so I am reserving judgment. I mean, like everybody else, I do agree that it does seem to have a short ilium, not just the ilium, but also the lower ilium, which is biomechanically important. It is not clear to me what exactly is going on with iliac flare. I think that's really hard to see from the casts, and I haven't seen the originals. I also haven't seen the models. It seems like the ilia do seem to be a little bit more laterally positioned, which is also important for bipedality. How much of that is actually there, I really have no idea. A lot has been made about the long ischium and what that has to do with function of the hamstrings. I'm not so sure that is quite important. It is true that bipeds have a short ischium. I don't know how detrimental it would be to have a long one. I really think the most important feature of the pelvis is the ilium, and I wish that I could see it.
Yeah. Just ask. I'm sure you can.
Yeah.
Could I speak to the question of the ischium? It is true that Ardipithecus does have a long ischium, and it hasn't been shortened and repositioned as it is in Lucy and other hominins. But you know, that is a great anatomy for a climbing animal. We know we know from electromyographical evidence in climbing primates that they are propulsive organs along with the quadriceps and moving up branches. Yes, and the dreaded vertical climbing that apparently Artie was not allowed to do, I'm sure it was capable of climbing vertically and obliquely, and the hamstrings would certainly assist in that. So I can sort of understand how you might have a stepwise process in pelvic modification, with the lateral balance and center of gravity issues sort of coming first. And I agree with you that for the kind of biped that Artie was, a long ischium probably was not any disadvantage.
Bill, could I ask you a question about the orientation of the iliac blades? The Artie reconstruction put out by Tim and Owen and their co-workers has the iliac blades rotated around so that this upper blade of the pelvis faces more laterally than it does in other early hominids, in the later ones, in Australopithecus, where it faces more or less backwards as it does in apes. And looking at that reconstruction, I can't help but feel that the position of those blades is to a very large extent dependent on the size and shape of the sacrum, and there isn't a sacrum. And if you put a narrower sacrum in there, a more ape-like sacrum, I think you would wind up with much less laterally rotated iliac blades, and it would look more like what you'd expect to see in an ape pelvis. What do you think of that? You've seen the specimens.
Well, I think that they argued that the space between the iliac blades was constrained a little bit by the lengths and orientation of the pubic ramus and the ischiopubic ramus below, that somehow, and that would obviously, that wouldn't affect only the iliac blades, it would affect the shape of the pelvic outlet. I don't know what obstetrical implications that might have, but I think that's the reasoning behind it, is that they had to accommodate the orientation and bring the pubic symphyses together.
But in the reconstruction, the pubes are not in contact with each other, so the
there's a symphysis of some kind.
Yeah, but the symphysial region is imaginary also. Not the part that's there, but the part that actually contacted isn't present in the fossils.
You mean the soft tissue?
Well, the soft tissue, Owen would say it's the epiphyses in between the ischiopubic bodies.
Yeah, again, I can't, I wasn't there but I think that's their reasoning. That's all I'm saying. I think is they were trying. They felt that was the most logical orientation. But I think as we talked about earlier, by your estimation, at least based on that picture, the sacrum of Artie was even wider.
Mm-hmm.
Than Lucy, which is remarkably wide to begin with.
Yeah. Let me ask a question, if I might, about the intermembral index. Okay. The story is that Artie has basically the forelimb is the same length as the hind limb. And this is being taken as a signal of a persistently primitive trait, a signal of a quadrupedal ancestry. Why isn't it a signal of incipient bipedality and an increase in the size of the hind limb or a decrease in the size of the forelimb? In other words, why is this a primitive trait rather than a derived Australopithecus-like feature?
Well, they argue that, and then it's very hard to do this for earlier Miocene apes, but they argue that they have proconsul, and so they feel that the proportions they see in Artie are more like that than they are of any great ape. I think that's.
Yeah, they are rather like.
It is also the case, and that what I find fascinating is that certainly the humerofemoral index of Artie is pretty much spot on Australopithecus afarensis, i.e. Lucy, which seems to be a perfectly fine index for a biped. So again, I'm not so sure that is a signal of pronagrade quadrupedalism. And I think we might even be agreeing on this, Matt.
Mm-hmm.
Questions? Yes.
Thanks. My question actually follows on this point. So Matt expressed some skepticism about there being sort of legitimate signals of pronagrade quadrupedalism in the postcrania. So, and you sort of had suggested that there really were some that were diagnostic of pronagrade quadrupedalism. And
I don't think there are any.
Okay. So is no one convinced that it was a pronagrade quadruped?
I'm not sure I'd go that far.
That's a tricky question.
You touch on a sore point.
Yeah. I think as most people read the document, and that's just so chock full of information, it takes a while to get through it. My interpretation, and I think Jeremy and I were talking this morning, he also came away with the same inference, and a lot of people I know thought that they were arguing that Artie, when in the trees, was a pronagrade quadrupedal palmagrade animal. And I didn't see any evidence of that in the hands in particular. But I've, in some interesting exchanges with Dr. White, he claims that was never said about Ardipithecus, that it referred only to the inferred locomotor adaptations of the ancestors that preceded Ardipithecus. So a lot of us have misunderstood, I think, what they were saying about the locomotor repertoire of Artie in the trees.
So I want to take a step back, I guess, and simplify this a little bit, at least in my mind. Could you guys take us into a brief sort of day in the life of an Artie? How often is it up in the trees? How often is it down on the ground and bipedal? Can we know that? When it is bipedal, is this a boreal hand-assisted bipedalism, or is it on the ground? And if it's on the ground bipedal, why? Because bipedalism is slow, and why would it do that? What is the environment like? What was it eating? I know there are a host of questions, but reconstructing this animal and fleshing it out a little bit, I think, would help us, if you can.
This is a tricky one. Obviously, I don't think we can get to percentages of time spent in the trees versus the ground using these fossils. They don't come with that kind of information attached. But it is an animal that spans that is critical in the human evolutionary story, because it is the first one that spans the gap between an animal that was selected partly for living in the trees and selected partly for living on the ground. In my view, and now Bill and I will speciate at this point, but in my view, the anatomy of Lucy afarensis shows where that selection was trending. It was trending away from the adaptive value of life in the trees towards adaptive value on the ground. So if we put Lucy on one side, and virtually all of the primates except the really large ones on the other, you can see that this is a creature that spends significant amounts of time in the trees. And that's what its foot tells us, and its lower pelvis. And potentially, and tied to the foot, tells us. So a day in the life, feeding, sleeping in the trees, moving from patch to patch on the ground, perhaps, and that's the link, basically.
I'm not hearing foraging on the ground and bringing back food to the females.
Again, are we talking?
I know.
Are we
Well,
talking
let me-
about the males or the females?
Let me jump in here and say that, I'm happy to tell you what I think. I'm less happy to interpret what my colleagues think who aren't here. So I long ago gave up reading those kinds of tea leaves. So that's what I think.
Well, all right, fair enough, but I mean, it's not as though Professor Lovejoy has not, in fact, expressed an opinion on
No,
this.
but Professor Lovejoy can speak for himself. He doesn't need me to do it for him.
That's fair enough, of course.
Well, I don't mind being wrong. So I think that Artie came to the ground and walked bipedally because she could. And by that I mean, I think I'm moving towards a position that bipedality may have co-evolved on the ground and in the trees. That I'm not talking about the orangutan model that Thorpe and Crompton have promoted, but something similar, more similar to what Sanu and Pickford have said, that truncal erectness, even some suspension, maybe assisted bipedality. I think Artie, since I don't find a pronograde signal in the hand, I'm thinking more and more that a lot of time was spent bipedality in bipedal postures and orthograde postures in the trees. So it came to the ground, presumably to forage. I don't know if it came to the ground to provision, males provision. I don't know about, but I am partial to models for origins of bipedalism that involve carrying. I think those, again, we're now creating, scenarios. But I prefer that model to standing over, looking over high grasses, or permanent estrus displays and crazy stuff like that. So, again, I think that I think the bipedal signal may be stronger than I thought.
Bill, has the mixture of arboreal and terrestrial adaptations in Ardipithecus caused you to revise your thinking about the importance of arboreal behavior in early Australopithecus?
Well, it's interesting that after a paper appeared that Jeremy was a co-author on about climbing in modern humans, Owen went on record to say that he always thought Australopithecines could climb, just that it was not sort of the direction of evolution's arrow, adaptive arrow. And I can agree with that. I think there's no doubt that afarensis was a more efficient, more capable biped. I mean, if they're right about Artie, Artie doesn't have the sort of knock-knee morphology that we see in Australopithecines. Its kind of bipedality would be ungainly by comparison to what I think occurred in afarensis. But I still think that those elements that contact the substrate, like the hands and feet, they still manifest features in Australopithecus that I think are not by chance being retained for a long time. I think they are enabling and reflecting an ability to get in and out of trees in a way that modern humans, which ain't bad, they could do much better than them. So again, I think my position, has, I'd like to think, shifted subtly, but I still think that Australopithecines, and some species more than others, probably still were involved in a good deal of arboreal activities, despite the fact that they were better bipeds than Artie.
I would just say that the argument about Lucy's locomotion, I don't want to get too far off on a tangent here, was never about whether Lucy could climb a tree better than we could or what have you. It was about whether her anatomy, her locomotor anatomy, reflects selection for current utility in the trees, or whether it is their primitive retentions and so forth. So no one ever said Lucy couldn't climb a tree. And no doubt she could climb a tree better than most of us in this room. But that hasn't been the critical question.
So we have questions out there. Alex?
Hi. My question concerns the cranial base. And first off,
Finally.
yeah, finally, sorry. First off, I don't really know, but are there, what is the cranial base record in the Miocene? In the earlier Miocene, are there any? And I think it's a pertinent question, especially for those apparently very orthograde suspensory European Miocene names. What would cause the cranial base to be reorganized for bipedalism as opposed to what seems to be very obligate orthograde suspensory activities with no real terrestrial quadrupedalism?
Right. There are very, very, very few clues from the Miocene as to cranial base anatomy. There are bits and pieces. There is the Ardipithecus cranial base, which, for my money, is essentially useless and is not reconstructible. My take on the cranial base reorganization has less to do with the posture of the trunk below the neck than what's going on at the level of the neck. For the following reason. If you look at gibbons, for example, gibbons are as orthograde as humans are, yet their neck posture is not that of a human. Their heads are slung forward on the cervical vertebral column, and the few data that are available on neck posture show they basically have the same neck angle as a chimpanzee. And their cranial base reflects that. They're orthograde, but they do not have any of the human modifications in their cranial base. So one of the gaps in the fossil record, it's a huge gap in our understanding in Australopithecus even, is what does the cervical vertebral column look like? And we were talking about this earlier because we don't really have any idea. We don't know whether a lumbar lordosis, which they do have, implies a cervical lordosis. We just assume it does because this is the way humans are organized, but the fact is we don't know. And there are a few cervical vertebrae from the Hadar collection of Australopithecus afarensis, and they show an interesting mix of human-like and ape-like characteristics. And so do the occipital condyles, by the way, in spite of their relatively forward position. They have a very strongly angulated articular surface, which is reflected in the mirror image of the C1 that we have, and the lower cervical, though not like an exaggerated, not as exaggerated as in chimpanzees. The spine is long and straight rather than short and inferiorly angulated. So I would love to be able to see more about the cervical vertebral column of Australopithecus, and maybe with some of the new material coming out of South Africa, we'll have a better idea. But at least in terms of afarensis, we don't. And I suspect there's some information there on the cranial base.
Speaking of South Africa, how does Mrs. Plez fit in here? As a lot of people have pointed out, STS-5 has a rather ape-like, long cranial base. The axes of the petrosal axes are oriented more anteroposteriorly and less transversely than they are in, say, robust Australopithecus, which is very human-like. What's going on with Mrs. Plez? How come she looks so ape-like?
Well, I would contest the description, first of all. I don't think that the cranial base of A. africanus, Mrs. Plez, is all that ape-like. Mm-hmm. The entire brain case of A. africanus in relation to most overall measures of cranial size is narrow. And that is reflected in some of the dimensions, relative dimensions in the central part of the cranial base. But the length of the base, no matter where you judge it, is short. And the relative to a chimpanzee, those landmarks in the middle of the base are spread out. And so, yes, it is a little less derived than the robust Australopithecines, which is, as you said, are a little more human-like in this respect, but it is by no means easily mistaken for an ape. Whether you look at the angulation of the petrous or in the relative dimensions of the landmarks under there. So I think it's an exaggerated, it stems back to work that Chris Dean and Bernard Wood did in the 1980s out of Chris Dean's dissertation. And they usefully pointed out the distinction, but it doesn't make them like a chimpanzee.
Bill, one of the things I'm really struggling with, and this is for both of you actually, that if these papers are right, chimpanzees are highly derived. And yet, when we compare these fossils to any, to the obvious things to compare them to, all we can do is compare to a modern chimp. And yet, if it's a derived animal, aren't we sort of chasing our tail here and saying, well, it doesn't look like a chimp,
Chasing the
therefore it's not a
absence of our
chimp.
tail.
Yeah, so how much, right, the absence of the African ape fossil record, how much of that is just really clouding our ability to know polarity of characters and to know any of this stuff?
It is, and what it's forcing us to do is to ask questions that may not be meaningful in terms of the answers we'd like. If we had a fossil record of chimp and gorilla evolution, we would be in a different position here today with regard to understanding Ardipithecus. But the fact is we don't. So, I like to say that in some sense we are like Charles Darwin in that in where we have a gap in the fossil record, we're forced to triangulate back to common ancestors from terminal taxa, and whether you're talking about humans or chimpanzees, or whether you're talking about Australopithecus and Paranthropus, at a given time slice, they are terminal. Mm-hmm. And so we are always, without a relatively complete fossil record, we're always forced to do the thing that Darwin did to great effect in the 19th century, which was to take chimps and humans and subtract the unique features of each and try to explain the common ancestry. Or in the case of humans, to project ahead how you would account for the catalog of differences. Mm-hmm. But of course, without a calendar of events, which is what the fossil record supplies, we have no test, except simply moving further down the cladogram, but it still doesn't help you understand the acquisition of the unique human characteristics.
So how would you identify a chimpanzee fossil in that case, a 5 million-year-old chimp? How would you know that's what you had?
Well, in the skull, I would look for relatively enlarged incisors, certainly, because they are the largest incisors. They and orangs have the largest incisors of any hominoid, extant hominoid. I would look for the exaggerated lower facial prognathism that chimps have that bonobos don't, for example. Okay. So forth and so on. Those are the sorts of things that I would expect to see if I were going to be certain I was dealing with an ancestral chimp, as opposed to something that was close to, at the common ancestry of gorillas and chimps, or was more closely related to gorillas, for example. But, this is just, hand-waving, because, we don't have the record, and we won't have it in our lifetimes.
I would hope to help diagnose it if I had a hand. I think if knuckle walking was a primitive condition, the last common ancestor, I think you could probably diagnose that. And because I think that that's a derived morphology. I used to think that it was impossible that chimps and gorillas had evolved knuckle walking independently, but I think arguments have been made that persuade me to at least consider that possibility as very viable. When I talk about the team of really insightful young investigators that I work with, I mean, we don't all agree completely. I think that Kaylee Orr believes that the wrist, for example, of Artie is very stiff. In fact, early hominins are very stiff, and he thinks that you can't rule out knuckle walking ancestry. But I guess I'm dodging your question. What would be diagnostic would be things that we already know occur in a chimpanzee. There might be entrapment of the sacrum in an elongated ilium.
Yeah, but you'd need to exclude the gorilla, though, too.
Yeah, that's it. Yeah, again, pick your homoplazy.
Yeah.
Okay, Eve?
Hi, so you've all sort of addressed the complexity of the phylogenetic picture before Ardipithecus. I'm sure a lot of us are interested in the picture that results after. So I was wondering how you all reconcile the idea that Ardipithecus leads into afarensis with when you consider the two potentially very different pedal morphologies, the two different feet around like 3 million years ago.
The Bertelli foot is what you're referring to, yeah.
Well,
I don't do phylogeny.
Chicken. Let me start out by saying that in the science papers of 2009, the lead paper by Tim White and colleagues presented as one hypothesis of three that Ardipithecus was a lineal ancestor of Australopithecus. I don't know, frankly, how committed he is to any one to the exclusion of the others, but Ardipithecus was entertained there also as as a late representative of a population that split off closer to the chimp-human divergence, and then evolved in parallel once that happened, or not in parallel, but evolved alongside, if you will, the lineage that gave rise to Australopithecus. So we don't know. We do know that the earliest Australopithecus at 4.2 million, Australopithecus anamensis, if, this is a big if the proximal tibia from Canopoi is associated with the craniodental material attributed to anamensis, then anamensis, like afarensis, was a terrestrial biped. Then the Bertelli foot would be a second lineage. It is also possible, because it's been published, that the Canopoi tibia may not be contemporaneous with the craniodental material from that site that's attributed to anamensis. In which case, one alternative is that, maybe anamensis is the taxon or the lineage, represent the lineage that's also the Bertelli foot. But these are hypotheticals at this point, and they raise questions, which is great, but at the moment we can't get very far into it because it may never be known for certain whether the Canopoi tibia is contemporary with anamensis or not.
Could I ask a parallel question? Given the existence of the Bertelli foot, what specimens do you feel most entirely comfortable, aside from the type specimen from Laetoli, assigning to Australopithecus afarensis, given that we have reason for suspecting there are two lineages in there?
I'm sorry, say one more time?
Yeah. What specimens do you feel entirely comfortable do represent the same animal as the type specimen of Australopithecus afarensis?
You're talking about the Hadar sample now. Are you asking me whether I think the Hadar sample is potentially divisible into two based on the presence of the Bertelli foot?
Yeah.
Oh.
Well, I wasn't. I was asking it in a less embarrassing form. I was asking what specimens do you think are entirely? What specimens do you feel confident pointing to and saying that's Australopithecus afarensis?
Anything with cranial and dental material, because that's how the taxon has been diagnosed. And that's how all the Australopithecus taxa have been diagnosed. None of them, to my recollection, have been diagnosed. Maybe Sediba is different, but no, I can't remember the diagnosis off the top of my head, but most, if not all, have been diagnosed exclusively on craniodental material. So if I look at the craniodental material from Hadar, I don't see evidence of diversity there. Variation, yes, but not diversity. If we get craniodental material associated with a foot or other postcrania that suggests that there's a lineage in the middle Pliocene subsequent to Ardipithecus that was spending time in the trees, then that, of course, then becomes an open question again. And this is, referring back to the late 1970s and early 80s, there's always been an opinion, vocal minority opinion, that the Hadar material is, quote, too variable to be accommodated into a single taxon. I've never been happy with that argument. I think it fails on statistical grounds, but and other morphological grounds as well. However, I will say that if it turns out that the Hadar assemblage does contain more than one, it will be very difficult to disentangle them, because in most respects, if they are sisters, they're going to resemble each other in far more ways than they differ. and it'll be a challenge. All I can say is that so far, none of the postcrania that's come out of Hadar, in my opinion, is redolent of a foot like the Bertelli foot.
Yeah, I agree. Other questions? Yes, Ed.
So, that's weird. In regards to Ardipithecus, I was just curious. I don't know how big she is, but wouldn't its size play a role in how much time it could be spending above the branches?
That's a very good question. 50 kilograms is a pretty big arboreal animal, and I think that the reconstructed body mass is potentially correct. I think one could make an argument that it might be a few kilos less, but it's still going to weigh in at over 40 kilos, I think, no matter what kind of regression or which bones you pick. And very clever people like John Napier long ago pointed out that, you get a big animal on a fairly small substrate, the stable position of that animal is not on top of the branch, it's underneath it. I think that it's certainly possible for a fairly large-bodied hominoid to be very arboreal, and we have plenty of examples. Orangs are a good example with extreme specializations. Chimpanzees are very arboreal, and there are different populations that vary in their commitment to arboreality. So, I don't think it precludes being a highly arboreal animal. And if you go to western lowland gorillas, with males that are huge, 165 kilos, that are capable of climbing with great facility, occasionally suspending themselves. I don't think 50-kilogram hominoid is out of the ballpark for a largely arboreal animal.
Alex? Yep.
So Professor Cartman talks about how you're not really convinced that human ancestors didn't engage in suspensory locomotion. And we also talk about that RD did not engage in that, and that Tim White's team believed that the ancestor to humans did not do that.
The common ancestor of humans and chimpanzees.
Exactly.
Yeah.
Exactly. But what would lead him to say that, given the RD's bones, and what would be his motivations to saying that our common ancestor with chimps did not have that, given that we talked about in class, Professor De Silva, that usually anthropologists and anyone actually has motivations in saying and having certain claims. So what would be their motivations for that?
Well, it turns out that I had the same experience that Professor Jungers did, except not with Tim White, but with Owen Lovejoy. That is, I went and said, wait a minute, you said that Artie was an above-branch quadruped, and he said, no, we never said that at all. I don't believe that. Okay, fair enough, I misunderstood you, and it's true. I can't find anything in the paper that exactly says that, but there's a one gets that false impression easily, but it's a false impression, fair enough. But if Artie wasn't an above-branch quadruped, then it was presumably doing a lot of suspensory kinds of posturing and locomotion. It was, as you would expect a 100-pound animal to do. It would have been spending a lot of time distributing its weight with the upper limbs under tension and the lower limbs maybe under compression. So the question is, given that Artie looks like that and that all the living apes do things like that, why do you conclude that the last common ancestor of Artie and all the living apes didn't do that? And the answer has to do with such things as the intermembral index, which resembles proconsul, and it does, fair enough, I believe it, I'm a convert. It has to do also with certain details of the anatomy of the wrist that Professor Lovejoy interprets as being indispensable to suspensory kinds of locomotion and that he does not see in Ardipithecus. I'm just not enough of an expert to speak to that. Perhaps Bill Jungers has something to say about it. Do you see a good reason for seeing in the Ardi skeleton reasons for saying that the common ancestor of Ardi and the living apes was an above-branch quadruped.
I don't, again, I think that it depends if you pluck almost any earlier Miocene ape out of the fossil record, you're going to be hard-pressed to find a suspensory adapted animal with the possible exception of Ardipithecus. So if that's your hypothetical ancestor, then that doesn't appear to be the primitive condition. But I agree with you there. I use a term maybe as a default. If it's not pronograde, then what is it? And it's anti-pronograde, which is a wonderful term that encompasses all kinds of arboreal behaviors, including climbing and suspension, anything pretty much except up on top of a branch in a quadrupedal posture. And I think that Ardi was capable of, if you want to put it in a quadrupedal position, I see it as more of a quadrumanous clamberer, where it's using gripping hands and feet to pull it along substrates. But I don't think there's any evidence that it was walking like this on top of branches with the hands flat, as a true palmograde monkey would have done. So.
Something that has to be said here generally is the Barcine and Star Wars issue. That is to say, there's general agreement among people who are very knowledgeable about the postcranial remains of Miocene apes that once you get past the sort of bottom stratum represented by things like proconsul hessaloni, that you're getting mixtures of features that are not precisely replicated in any living primate, and so it's not clear what these things were doing. And when we start applying categorical locomotor terms like brachiator or above-branch quadruped to these animals, we're almost certainly doing injustice to the complexity of their locomotion. I just think that there is a lot of tension in the upper extremity going on in these animals, which is not the case in typical quadrupedal monkeys. That would be the most radical position I would care to articulate in public. And I gather that there's a general agreement on that. And I think that Professor Lovejoy would say that about Ardipithecus. I'm just puzzled by the fact that he does not want to say it about the common ancestor of Ardipithecus and other hominoids.
Other questions? Christy?
I'm relaying this question from afar. Not afar, but afar.
From the afar.
Syllables, they make such a difference.
This is actually from Kalior. Is a generalized Miocene ape like proconsul really a good outgroup to hominins? Couldn't supposedly shared characteristics of hominins and some Miocene apes be homoplastic rather than simplesiomorphic?
Again, it depends on what part of the body I think you look at. Again, I think in terms of femoral anatomy, it is the Miocene ape-like proconsul is remarkably more similar to a hominine than it is to a great ape. But rephrase the question again. Is that would you restate?
Homoplastic.
Yeah, is it possible? I guess it's possible. I'm not sure how one would answer that. Again, similarity seems to be the strongest case I can make for certain parts of the Miocene apes, making reasonable ancestral morphotypes for later hominins, better ancestral morphotypes, if you will, than any of the living great apes.
I think this is perhaps part of a larger question, which is if there has been evidence that extant African apes are homoplastic. So if we accept that chimps and gorillas are homoplastic, then why don't we accept that any shared similarities between Miocene apes and hominins are also homoplastic? I think that's what he's getting at.
I mean, there's no limit on the amount of homoplasia you can invoke. There is no practical limit. Anything is possibly homoplastic. But if we start at the position in trying to reconstruct the shape of the tree of life that anything goes with regard to homoplasy. We might as well stop because we can't get anywhere. Homoplasy is a wonderful thing for functional anatomy because it gives you general cases so you don't have to rely on unique ones if you can find them. But it's not a starting point for phylogeny reconstruction. It's not a tool for phylogeny reconstruction. It's an outcome of phylogeny reconstruction. And if we don't adopt that as an operational principle, we can't reconstruct phylogeny. Hennig called it his auxiliary principle, that derived similarity is assumed to be homologous unless you can show the opposite. And David Wake was quoted much more recently than Hennig as writing that one doesn't search for homoplasy. Homoplasy finds the researcher. So homoplasy, yes. But if we let it in the front door of analysis at the outset, we can just sneak out the back door and go and do something else. I'll become a functional morphologist. Well, maybe I won't. But
No, stop, please. Could I put in a slightly more skeptical position than that? I think there are problems with the identity. I agree that homoplasy emerges at the end of the phylogenetic analysis. But I also think homology emerges at the end of the phylogenetic analysis.
yeah, but not synapomorphy.
Which is another one. No, I think, okay. No, I think synapomorphy emerges at the end of the phylogenetic analysis. And I think basically what you're doing when you do a phylogenetic analysis is you get a list of all let's just take the simplest case, 3 taxa: human beings, chimpanzees, donkeys. Let me pick a better example. Human beings, chimpanzees, goats. Okay? And you make a list of all the resemblances. All right? And then you try to decide which resemblances are synapomorphies. Those are the ones that count for phylogeny. And one synapomorphy is sufficient to establish what the sister groups are. So your problem is determining which of these clusters represents the true phylogenetic signal. And the usual way to do it is something like counting. Okay? There are signals of all 3 possible kinds. We could say that human beings are more closely related to goats because there are beards in the males in both species. Okay? We could say that chimpanzees and goats are more closely related because they are covered with fur and human beings are not. Or we can say that human beings and chimpanzees are more closely related. And it turns out that if you look at all of the similarities that human beings and chimpanzees have in common with each other, they all turn out to be most of them turn out to be plausibly interpreted as either synapomorphies of humans and chimpanzees or as simplesiomorphies, primitive retentions like having 5 digits on each hand instead of 2 that have been lost in the goat. Okay, but this is basically a process of weighing the 3 piles and seeing which is the biggest. And there are other ways of doing it, like Bayesian probability, that I don't really understand, but they amount to the same kind of thing. You're making an estimate as to which of the 3 possible assumptions is the least improbable. And
obviously, treating characters as uncorrelated is a problem for functional morphologists.
Absolutely.
Many of those characters that you put on the list, is that a giant character complex that counts as a heavily weighted one character?
Absolutely.
Or is that, or do we count them all independently?
And there are two kinds
of philosophies.
There are 2 kinds of correlation that have to be taken into account here. One is functional correlation. Okay, in general, you are not going, for example, to find redirection of the occipital condyles without movement of the foramen magnum in a corresponding direction, because things won't work if you try to change one of those and not the other. Then there are genetically correlated things where a single genetic complex affects developmental changes in a whole bunch of pieces of morphology because of the way that ontogeny proceeds. Professor Lovejoy thinks that a great many things about human evolution can be explained in those terms, whereas functional morphologists tend to think, no, these are all separate pieces that are independently subject to natural selection. So there's a good deal of debate about how things ought to be weighed, how you count things up as independent items to be counted in the balance, and so on. But so there are a lot of difficulties about doing this.
Absolutely.
And,
Oh, yeah.
But do I, is it possible to do it? Yes, as I was saying to our distinguished visitors before, during the interim, there's no question that we know a lot about phylogeny. Okay, here's a 3-taxon tree. Human beings, donkeys, broccoli. Okay, I have no doubts about that. It's just true, period. Okay, but human beings, Ardipithecus, Australopithecus anamensis, we don't have enough knowledge to be sure.
Yeah, and I agree with everything you say. And my friend and colleague Bernard Wood, who's very concerned about homoplasy these days, he expresses it as he did just a week ago to me, that homoplasy is a concern because it prevents you from having too much confidence in your hypotheses. Right, so you need to be prepared to jettison them, and that's fair enough. And from my point of view, thank goodness I've not had any hypotheses refuted yet. So I'm still waiting.
Other questions?
With more discoveries, obviously more light will be shed on Artie's actual location on our phylogenetic tree. In your opinions, what is the best anatomical trait that they could find that would give the most definitive answer about whether she was ancestral of humans or a completely extinct side branch? What discovery would shed the most light on that?
Ooh, Christmas wish list for Artie. Yeah.
I'm sorry, there was one little component of that I didn't quite get, which was near the end, which...
What would you wish for, I think, if in a new discovery
Yeah, for an Artie.
to help resolve some of these issues?
What could you find in the fossil record that would shed the most light on the issue that we're discussing today? What would elucidate the most as far as anatomy found in the fossil record?
I'd like to see a vertebral column.
Yeah.
A vertebral column
Yeah.
would be really nice. The femur of Ardipithecus would also be nice.
Scapula.
Scapula, vertebral column, sacrum,
Calcaneus.
the back of the cranium.
A 6 million-year-old gorilla.
Yeah.
The 6 million-year-old gorilla in the room.
Kim's already found that.
I mean, so there's plenty that's still unknown about Ardipithecus, as there is about Australopithecus, the cervical vertebral column, for example. So there are all kinds of gaps remaining to be filled. But the list obviously depends on which particular question you're interested in. If you're interested in whether it's a hominin or not, there's a certain list of things you'd want. If you're interested in reconstructing the last common ancestor, that's a different list.
We keep going around saying, if only we had some more fossils. And every time somebody finds some more fossils, it's another damned species. Okay? And that, not every time, but frequently enough, the Bertelli foot, the Sediba material, who knows what we're going to be getting out of the Rising Star Cave in South Africa. And every time you add a new species, you add an appallingly large number of possible cladograms to the alternative cladograms to the system. What we need is fewer species. That
You
would
can start sending stuff back, I suppose.
But it also depends on the kind of question you want to address. I'm comfortable with Artie being in the hominin club, but I would be hard-pressed to make a case for it being the ancestor of any specific species. Those situations are so rare, and I guess by good luck and good looks, you were able to find that for Anamensis and Afarensis, but that's such a rare phenomenon in the fossil record.
It is. It's extremely rare.
well,
In the hominin.
it's rare in terrestrial mammals.
Yes, indeed.
Okay, it's not rare in, say, marine invertebrates.
No, right, that's correct.
I hesitate to say that because otherwise some creationist out there is going to stand up and say, see, there are no missing links, just as we said.
Not a big missing link.
Other questions? Yes. Caitlin.
I think this was addressed earlier a little bit as to why would Artie be bipedal, kind of like in her daily life, what would she be doing? And I know someone said it would be kind of moving from patch to patch, from tree patch to tree patch, but from my understanding, she evolved in a more forested environment, so I don't know if that's wrong or not, but I thought she was more in the forest, so why would she then like struggle with being half a climber and half a biped? Because if you're not really good at either, wouldn't you just be like
Well,
eaten?
chimpanzees have faced the same problem and just solved it in a different way. I mean, they're great arborealists. They're also very good in terrestrial locomotion. And even if you're an arboreal animal and build nests and forage, you may exhaust the resources in that tree, and you have to come down in order to ascend another one, even if you're in a forested environment. So I don't think you need grasslands in order to invoke the value and benefit of being a biped.
There's also a minority opinion about the environment.
But part of that difference of opinion also depends on different definitions of what a savanna is.
Yes.
And I want to say, first of all, that nobody has suggested, to my knowledge, that Ardipithecus evolved in or lived in a tropical forest like you find gorillas and chimpanzees in, not including some of the so-called savanna chimps. They lived in a woodland, and it may have been more closed than open, but woodlands differ from forests in the degree of canopy continuity. So woodlands, you can move from, even closed woodlands, you can need to, if you're an omnivore, you move from patch to patch in ways that, gorillas don't when they, in the tropical forest. So the definition of the habitat or the environment in which Ardipithecus resided needs to be very carefully characterized, because it's easy to get the wrong impression.
I have another question for you guys, and in fact, I hope I can get some feedback from the front row there with Professors Knott and Langergraber as well. In publishing these papers, one of the arguments made was that chimpanzees no longer make a really good model for understanding early human evolution. And I suppose the first question is do you agree? And if not, or even if so, what role or what value do studies of chimpanzees have for understanding human evolution? And saying it to folks who study the fossil record, and then saying it to folks who study living chimpanzees and living orangutans as well. So what do you think?
Well, just because chimps are our sister taxon does not grant them a special place in our explanations of how humans became unique. Obviously, there has been evolution that we don't know about in chimps, but obviously there's been 6 to 8 million years of evolution along the chimpanzee lineage, just as there has been in humans. Mm-hmm. But I don't give chimps a pride of place from an anatomical and behavioral point of view in trying to understand how humans became humans. Absent including gorillas, orangs, gibbons, monkeys, and many other primates and many other non-primate mammals. I resist the temptation to engage in using terminal taxa as living ancestors. And I think we are led down, garden paths that way. We have to be very eclectic in the data we utilize to understand how humans became humans, and that means casting a broad net on the tree.
It's nice to have other facultative bipeds, which chimpanzees are, and to understand how their musculoskeletal system either facilitates or limits the kind of motions they can make. I would assume that chimpanzee dental morphology and isotopes, would also be informative, perhaps, about.
But if chimps had teeth like gorillas, I don't think, we would be any more inclined to use those teeth, or any less inclined to use those teeth.
Well, maybe not for maybe not for phylogeny.
No, that's what I'm saying. That's what we're talking about, though, I think.
Just for purposes of reference on the environmental issue, the quote is that Ardipithecus ramidus preferred the more wooded habitats along the available spectrum in the regional geography, that is ranging from closed canopy forests to open grasslands, and that this is a distinction between Ardipithecus and Australopithecus, and that, quote, the integration of available physical and biological evidence establishes Ardipithecus ramidus as a denizen of the closed habitats along this continuum.
Yeah, but closed habitats doesn't imply tropical forest, completely closed canopy. They were closed. They just did the closed. I don't think they're in Eastern Africa during the Pliocene. I do not believe there were habitats that look like the habitats that living apes are in today.
What about galleries?
There were no doubt galleries, but unless they were confined to the galleries, seems unlikely to me. They were on the ground, moving around from patch to patch.
Yes.
All right, so now that I've been disabused of my misunderstanding about whether or not Artie has ever been claimed to be a quadruped, you've said a couple of times, pick your homoplasy. So if we reconstruct, how much homoplasy are we picking if we reconstruct the LCA as a quadruped versus if we reconstruct the LCA as a suspensory animal?
I'll need an envelope to.
Good question.
Granted the complexities of knowing how to weight a character, integration, all that sort of thing. But just ballpark, your gut feeling, how much homoplasy are we picking?
Part of the problem here is weighting the traits. Okay, and nobody does this when they do formal phylogenetic reconstruction and crank the trait list through the algorithm. But everybody does it when they think about it. And for example, I think Professor Kimball would say that he regards it as much more likely that knuckle walking adaptations have been independently evolved in chimpanzees and gorillas than that the loss of the canine P3 honing mechanism has been independently evolved in Ardipithecus and Australopithecus. So you're making a judgment as to what you make in arguing these things. You make judgments as to what you think the likelihood is of things evolving in parallel. I didn't mean to put words into your mouth, Bill. Maybe you wouldn't make that judgment, but I was just suspecting that you would.
Well, no, I mean, I wouldn't make the judgment by comparing the one character to the one character, but with regard to Ardipithecus, at least, it's more than one synapomorphy. It's the canines, it's the cranial base, and depending on how you view the cranial base, possibly third character is the terrestrial bipedality. So they are independent, unless the cranial base is speaking to the bipedality, they are independent corroboration of one another, right? That's the strength, is that you have two, perhaps three independent systems that are giving you the same phylogenetic signal. That's where the strength comes from. Now you can turn it around and say, well, we've got knuckle walking, and then we've got the unusual supraorbital torus of the African apes, which you don't find anywhere else among the catarrhines. And all other things being equal, they're great synapomorphies, but they're not. I mean, they are synapomorphies, but they're not homologous, right? They're not homologous. So you could, ultimately it becomes how big is the pile, right? Because I can't see of an objective way to say that in the skull it's more likely, homoplasy is more likely than in the postcranial skeleton. And there have been arguments both ways, but the most convincing ones that I've seen says that even characteristics within the masticatory apparatus and other functionally sensitive areas of the body, which since Darwin have been held up as the least likely to be faithful to phylogeny, it turns out that that's not necessarily true. So,
I don't think one system is more likely to be homoplastic. I think there's been some work on that. Didn't Blythe Williams, didn't she do a study of other mammal groups where the homoplasy was just as likely to occur in the craniodintal region as in the postcranium?
Yeah.
Wood and his co-workers, of course, have come to very different conclusions.
So you take your pick, basically.
Are there other questions? Ed in the back.
So if Australopithecus afarensis is after Ardipithecus and afarensis is much more bipedal than Ardipithecus, do you think Ardipithecus sheds any light on what the driving forces for bipedalism would be, since would have a lot of negative pressures, since you can't outrun anything, really? So why would bipedalism be selected for?
Seems like a good question to end on.
Yes, yes.
Well, I think maybe the type of habitats that afarensis is in, probably the frequency of bipedal behaviors. You know, I certainly would agree that afarensis is a much more terrestrially adapted hominin than Ardipithecus. I don't think, I think there is, I used to think that there was evidence of hind limb elongation that was modest in Australopithecus. Now Artie makes me less secure about that, because I know certainly by the time we get to Homo, you have a reconfiguration of the body plan with long hind limbs, and that's related to the economy of locomotion. But I think there really are features, like we talked about the big toe and Artie being sticking out like a big sore thumb. That's gone by the time you get to afarensis. I'd be fascinated to know if it's gone in anamensis. For me, if the Christmas wish list of fossils, I'd love to see more Artie, but right now I would love to see a postcranial skeleton of anamensis to help us understand if it makes a reasonable link functionally and phylogenetically between afarensis and Artie.
Yeah, and I actually agree that it's undoubtedly related to the increasing prevalence of open habitats. And if that is in fact the case, then actually we may see a functional link between the increasing incidence of bipedality and dietary change. Because the early Australopiths show a dramatic differentiation of the masticatory apparatus by comparison to Ardipithecus, no matter how you do it. I mean, if you look at the skull of Ardipithecus, except for the cranial base, the rest of it in the face and the dentition and so forth looks nothing like Australopithecus, except insofar that they share a more general set of similarities.
Great,
So
Bill.
it
I
may
think
very well-
we have to wrap up. We're getting to our end of time here. So if you have more questions, certainly come see us in the reception area afterwards. I want to thank Kay Brown for the idea for having this in the first place, for bringing these folks here, and for organizing all of this. Thank you so much. And nice job with the questions, you guys. You did BU proud. Very nice job. So thank you all very much. Thank you, Drs. Kimball and Jungers, and thank you, Matt Cartmell. Thank you very much.